Okay. Okay, let's get started. Try to record this session as well. Not minimizing. Just keep it like this. All right. So we are going to go over the data today. How many of you were able to download? Ah, good. Did anyone succeed in running it properly? Some of you, huh? Ah, good. So great. For the rest of you, we'll just go over that today really quickly. So let's get started. So we were talking about the data that we are going to use in this course. So we went over the power spectral density and time frequency plots very briefly. This is the meditation thing. And in the last class, we had just started talking about the type of data that we generally record from monkeys. So in our case, this is, we use Utah arrays. These are the Utah arrays. It's a 10 cross 10 grid that is implanted in the monkey's brain. And then when the monkey looks at these gratings, then you get these types of responses. So in this case, as you change the orientation of the stimulus, you get different gamma responses. In fact, in this case, you have a slow and a fast gamma. So this is fast gamma, this is slow gamma, which for some reasons we think indicate local and global processing. So this is where we had stopped in the last class. I'll set my alarm. 9:25. Okay. And then I told you to download this monkey resources folder. So I will show you step by step what what this data looks like for those who haven't done it yet. All of you should be able to do it by now. So let me just come out of this and explain a little bit about. Okay, so suppose I have this monkey resources folder that I have downloaded from the link that I had shared with you. So there are 3 folders. Actually, there's one more that I just added yesterday. I'll tell you about it. So this data folder contains, all the data. So we have 2 monkeys, Alpha and Kesari. So we had done one implant there with just the microelectrode array. So that then we used to call them Alpha and Kesari. But then we did another surgery and implanted a hybrid array. So to indicate that, these are called Alpha H and Kesari H. Let me see if I have. So this is a hybrid array. This is in this case, a 9 cross 9 array, not a 10 cross 10. So 81 electrodes in this array. And we also have this, what is called electrocorticogram. These are electrodes that are placed on the surface of the brain itself. And it's a 3 cross 3 grid. So these are Agrita is this, the main student who was involved in this project several years ago. And Dr. Sebastian was the vet who helped in this. So this is how it looks like. So this is the nose of the monkey. These are the ears. This is where the primary visual cortex is, back of the head. And we have implanted this 9 cross 9 array right in the brain. So this is the brain. And we also put this ECOG array. So we cut the elastic here, so so that we could put this electrode. So they were so this microelectrode is surrounded by 9 electrodes, of which these uh, 6 are in the primary visual cortex where we are interested in. Now, out of these 6, we were able to, I think, record from 5 of them. So this is how recordings look like in our lab. So I thought I'll show you a little bit. So what you see here are LFP traces, local field potential traces from 3 out of the 81 electrodes that we are, 90 electrodes that we are recording from. The monkey looks at a screen and we have a copy of the same thing on our screen. So this is whatever you are seeing here, the monkey sees the same in another room where the monkey is sitting. Now we have implanted this 9 cross 9 array 1 to 81 and that's indicated. So this is how it would look like in the brain itself. Okay. So these corresponds to this 9 cross 9 array.
What is the rough size of one of the edges?
Edges?
It's a square array.
So 400 microns apart. So about 4 mm by 4 mm. Those are some bad electrodes. Some electrodes are non-functional and you can't use them. So this information is also now provided to you. And in addition, so this is what you have in the brain. And these neurons only respond to a very small part of the visual space, which is called a receptive field. So actually, in this on this monitor, which is this is a copy of the monitor that the monkey is looking at. We actually just put a photodiode exactly where we have the receptive fields, so that we know exactly what stimulus is being shown at what time. So there is actually a very small part of the visual space. 0, 0 is the fixation dot. So in this case, the monkey is fixating here, and it's only about 3 degrees apart. And that's a beautiful retinotopy, which means that as you move in the brain space from 1 to 9, for example, so here from 1 to 9, the receptive fields shift in a very sort of regular way in both directions. Okay, so it's as if the whatever there is in the visual space is mapped very precisely to what you have in the cortical space. Okay. The ECOGs, those big electrodes, have larger receptive fields and they are also a little bit farther apart. So in this case, 82, 86, and 85, these are 3 electrodes. We also have 2 more electrodes, but they are so far out that we had to actually map it with a separate protocol. So there are 5 ECOG electrodes that are active. And let me just see if this runs. Okay. So you will hear this tick. That's where the trial starts. The monkey fixates. I'm not showing the monkey's eye data here, but the monkey will quickly fixate exactly at 0, 0. And then a series of stimuli, in this case of different colors, are shown. And just keep looking at these traces, and you will see that for some colors, you will see that the line thickens. So this is the gamma wave. And tell me when you find the strongest gamma, which color? And let it run for a while. That's a great thing. Not that strong gamma, but it's still there. Blue has gamma. That's the red line. Let's see. It's very clear. In raw traces, it's generally hard to see gamma. The grating actually produces very strong gamma also, but it's not easy to see in raw traces. But red colors induce huge gamma oscillations that you can in fact even see in raw traces. Okay. So when the monkey does the task correctly for 3-4 seconds, it keeps fixating, then the monkey gets juice as the tonak sort of sound. Occasionally, the monkey will just look here and there and not finish the task. Then there is another tone that's associated with it, and those trials are not counted.
So how do you know that the monkey?
So we have an eye tracker in the other room where the monkey is sitting. So we know exactly where the monkey looks at. So if the monkey eye moves away from 1.5 degrees of that fixation spot, the trial gets aborted. Of course, no juice then. Okay. So now let's get back. So the monkey would do this task, let's say for 2-3 hours. So you will end up showing lots and lots of stimuli. Then what you would do is every time the stimulus is shown, starting from that, you take a segment around that start point. Okay. And you collect all those, and that's what the data that I have given you looks like. So now I will just show you a little bit of the structure of the data. Let me just clear all of this. Okay, so let's say I go to alpha H. Microelectrode is the type of recording electrode. We also have EEG. So this is microelectrode. Experiment date or date is the date on which this was done. So this was 21st August 2017. Then the protocol name in this case indicates what kind of protocol is being done. So this is GRF002. And then you get to segmented data. There are lots of things. We'll only pay attention to LFP right now. So LFP, and let's look at, let's say, electrode 1. Okay, so I can just drag it here, or I can just say load this. Oops, now I can't drag. Sharing your screen. Yes, I am. But let my. Just change the country. So usually it's easy to just drag it. Never mind. So I'll just do it from here. Um, so I'll just go to monkey resources. Then what was it? Data, alpha H, microelectrode, this day, segmented data, and LFP. Okay, so I just change my directory there. So now I can see all the electrodes and I can just say load elect 1. So when I do this, you will get this variable called analog data, and it has a size of 1384 cross 4096. 1384 corresponds to the total number of segments that we have collected. Okay. So if I just say plot analog, is this me or someone else? Okay, so these are the 1384 traces, each of length 4096. And you might be wondering what time values these correspond to. So we also have a variable called LFP info here. So I can load that, and now it will give me this time vals also. So now I can just plot like this time vals. Now the x-axis becomes time. So time 0 is when the stimulus is shown. And around that time, I'm taking about a little bit more than 2 seconds of data. Okay. And the stimulus, as you guys saw there, was shown for a short duration. It was 800 milliseconds. So this doesn't make much sense, but let me just look at the average of all these traces along the first dimension. And this is what is called an evoked response or event related potential, ERP. Um, right after the stimulus, there is a negative dip, and there is something else that goes on. And when there is an offset of the stimulus, there's another this offset response. This one is the offset response of the previous stimulus. Okay, they were shown in sequences. So there was the stimulus is shown for 800 milliseconds and the inter stimulus interval in 700 milliseconds. So this is the off response of the previous stimulus, and this is the next one. Okay. Now, as you saw, different stimuli are shown to the monkey, right? So it's not the same stimulus. So you need to know what are the stimuli that are shown to this monkey. So for that, I have to go to extracted data. And there is a variable called parameter combinations. So I'll load parameter combinations. Oops. So the parameter combinations essentially is a cell array, and the visual stimuli that we are showing, in this case, it's a grating, it's not a color stimulus, is defined by 7 parameters. Okay. These are the azimuth and elevation that tells you where the stimulus is located in, azimuth and elevation. So these are locations in the visual space. In our case, it doesn't really matter because the stimulus is really large. But if you had a small stimulus, you had to tell where azimuth and elevation is. Sigma corresponds to the size of the stimulus. Again, it's not relevant for our case because we just put a massive stimulus. Then there is spatial frequency, which is the spacing between the white and black bars, the orientation, the contrast of the stimulus, and temporal frequency, which is the drift rate. So in our lab, we just run many of these protocols. These are called Gabor RF map protocols. That's where this term GRF comes from. And we basically just keep changing these parameters to generate different types of stimuli. Okay. So the data set that you have, now if you go to size of parameter combinations, it's basically a single azimuth and elevation and a single size because it's a large full size screen. So those we are not varying. What we are varying are 2 parameters, spatial frequency and orientation. Okay. And if you want to know, so spatial frequency, so it's F vals unique. These are the 5 spatial frequencies, 1, 2, 3, 4, 5.
Where the 5 values of the size of parameter combination?
I'll get to it. And then 8 orientations. Okay, so in this, we are showing 40 different types of stimuli. 5 spatial frequencies, 5 orientation, all possible combinations of those. So if you ask, okay, what are out of those 1384 trials that we have, which correspond to FVAL's spatial frequency of 0.5 and orientation of 0. So to get that, you just have to do 1, 1, 1. In this case, the first of each, 1, 1. And these 35 indices, these are the indices. So trial number 29, trial number 52, dada, up to trial number this one. These 35 correspond to stimuli when a spatial frequency of 0.5 and an orientation of 0 was shown. Does that make sense? So now suppose I want to look at the ERP of that. I'll just simply write parameter combinations 1, 1, 1, 1. And the second A is I want all 4096 values. So I write it like this. This is how it is done. And these are the ERPs for that one combination. Similarly, if I wanted to show for, let's say, the second orientation or the 90-degree one, I'll just change this to 0, 22.5, 45, 60, fifth one. So that would correspond to this one. So this is for 90 degrees and the other one is for 0 degrees. And so on. If you just keep varying these two variables, you will get for any combination. Now, why is there an extra one? Because the last one, so parameter combinations. This one had 35, so did the so do all of these. Sometimes there's one less. The last one is a combination of all the different orientations. Okay. So if you want to say, show me a response for spatial frequency, considering all the orientations together, then you would just use the ninth one, which will have, um, you know, um, 35 times 8 orientations. 35 times 8. What is 35? Yeah. So occasionally one trial is not shown on some years, so might be a fewer, a little bit less. So this is why whenever you have more than one variable, so 5 cross 8, the parameter combinations is 6 cross 9. Yes. Huh? What does every You are concatenating all the trials in which 0 or 22.5 or 45 were shown. Suppose you're not interested in the orientation difference, you just want to know how things are changing with spatial frequency, combining all orientations. Then you would use the all combination. Okay? All right. So now, once you have this knowledge, actually you don't really need this knowledge to run the GUI, but nonetheless, so let me just go back to so I'll just go back to monkey resources. Actually, the path doesn't matter. So suppose you have downloaded all your data on the desktop. So it looks like this. This folder sitting on your desktop with monkey resources. Within this, there are these what are called common programs, which contain a lot of different things for segmentation and display and all that. So it took us a long time to write this. You won't be able to go in and figure out all the codes. There's a very extensive coding that's there. But what you need to do first up is add that to your MATLAB's path. So there is this new environment set path option. So you have to just say add with subfolders and go to monkey resources and common programs. Sorry, go back. And then you just choose this and it will add those. It's very straightforward. Cronux also, that's another sort of software that we use. You should actually download it from their web page. This is an old version that I have. It's free software. Then you save, and then MATLAB knows the location of all the programs that we are going to use. Once you do that, you just have to use this command called display single channel GRF. Okay, show individual channels at a time. We have an all channel version also, which I'll show. And then it asks for the name of the monkey, then the experiment date, and then the protocol name. These don't change. What will change is the location where the data is kept. So depending on where you have kept your data, you have to change this string. In my case, it's kept in desktop. So I have given this string. You can give whatever C drive, comma, and monkey resources or wherever you have kept it. Yes, same commands. Mm. So I think I'll close all the windows and then I'll run this. Yeah. Then if you are lucky, then you will see that a big GUI like this will open up. Actually, it's a very stable code. So chances are that you'll be, oh, okay, there's some resolution issue. So we are unable to see this. I'll try to change my screen display. Okay, so this sometimes makes it a little unstable, which is why I usually don't want to do this. Okay, so now it's slightly better. So let's look at the data from electrode 1. Um, and this is sigma. It's irrelevant in this case, but all sorts of options here. So there are, you can see five spatial frequencies. So let's say I say show me for all, and again eight orientations. I'll choose all here as well. And I say just show me the evoked response, the ERP, and I do plot here. Then it shows me this is the average of the all conditions. Okay, so all 1384 trials. Here they are separated by orientation and spatial frequency. So there are 8 orientations. So this is all spatial frequencies with 0, 22, 45, 67, these orientations. Okay. Likewise, here this is all orientations and spatial frequency of 0.5, 1, 2, 4, or 8. So this is the evoked response. That doesn't look very different. If you look at the time frequency plot, so in this case, the raw time frequency plot will look like this. Remember, I told you there's always a lot more power at lower frequencies. As we go up, the power goes down. So it's a little harder to see in these plots. These are gamma waves. But if I sort of normalize this time frequency plot by subtracting the baseline response. Baseline means pre-stimulus response. By the way, those are also these parameters here that you can change and play around. In our case, baseline is minus 0.5 to 0, where 0 is the onset of the stimulus. And clear all the plots. And you can see, so this is the plot I was showing. See, this is the gamma response, fast gamma. It is the strongest for 90 degrees. It falls away. And you have slow gamma here, which sort of goes, it's a bit less here, and then it comes back. This is for one electrode. You can change the electrode and, play around and see what happens. There's a dependence on spatial frequency also. 2 and 4 produce more stronger fast gamma response. One actually produces both of them. So if I wanted to better see fast slow gamma, I could just say use a spatial frequency of 1. And now it will show me the orientations for that spatial frequency. If I plot this again, now you can see the slow gamma much better. It's tuning for that. Okay. Okay. So any questions on this? Now, how we are getting this plot, how we are making this, this is not a very, so we have much better ways of generating these time frequency plots. Those we will cover for the rest of the course.
What are those plots? The bottom two, the long ones.
These ones?
Huh.
Yeah. So this one just shows the plots for all combinations, size, orientation, spatial frequency, contrast, and temporal frequency. Because we are not varying, these three are essentially the same as that. In fact, there is an option if there are multiple azimuth and elevations that are shown, those get shown here in a sort of a two-dimensional matrix. So you can see how it varies with a location also when you use small stimuli, but we are not doing that. So everyone happy with this? Okay. So there's also an option to look at firing rate data. So let me just see if this electrode has any firing. So firing rate, let's check. Not great. Let me see some other electrode. Okay, it is responding. Spike rate is not that high. Actually, so LFP is very stable. Most electrodes will show a good LFP response, but only some electrodes will have a good spiking response. So let me just run this other program called display all channels. It's looking for EEG. So I need to tell that it's actually a micro electrode. By default, ah. Okay, so now this is the entire grid, 81 electrodes, and the ECOG electrodes are shown at the bottom. So the size, and now let me again do all here and see what I get. And in this case, I'll make it firing rate. So we are essentially asking which of these electrodes have nice firing. So many of these have a nice firing rate. So let me just choose this one. 16, 17, 18. Okay. I'll tell you what this is in a bit. I'll just go back and show you. I'll just make another one. And choose electrode 16 here. Actually, I'll show you raster plot first. Okay. Actually, let me just, it's a little complicated. So I'll just make this a single spatial frequency. Let's say. Okay, so what you are seeing here, this x-axis means a trial number in these plots. I'll tell you what this means. So essentially, the monkey saw the stimulus 35 times. So this is 1 to 35. Okay. In this case, every time a stimulus is shown, the neuron fires action potentials. That's how neurons talk to each other. That is indicated by a tick in each of these rows. So you see that as soon as the stimulus is shown, there are lots of these ticks. So the neuron fires a lot of action potential, but it's not coming at exactly the same time. There is some variability. So this is the classic neural response, and this is the classic tuning for orientation and spatial frequency. This particular neuron that we are recording from electrode 16 likes this 22 degrees stimulus because there are a lot more ticks there. And it also likes lower spatial frequency. So you can see that lower spatial frequencies produce a stronger response than higher spatial frequencies. So this neuron is tuned for a 22-degree orientation and low spatial frequency stimulus. The firing rate is obtained simply by essentially counting the number of spikes in some bin and making that as a plot. So this is how it would look like. It's tuned for actually 0 has an even strong response. It's a very nice tuning of this particular neuron for 0-degree orientation and this spatial frequency. If I look at some other neuron, let's say, well, 17, let's see what that neuron likes. So you see different neurons actually prefer different spatial frequencies and different orientation. That is how the neurons actually break down the visual space into, sort of like basis functions where you are essentially approximating them with edges of different orientations and different spatial frequencies. You can play around, look at the neuron units that have electrodes that have good units and what their tuning preferences. These are mostly multi-units. So the segmented data actually has a folder called segment that stores the spike waveforms. So you can do spike sorting and find, like isolated units if you don't do that, this is unsorted. Mostly you get multi-units. But generally the tuning is quite decent. So it must be one or at most a few neurons. Okay. How does the detect? So we are looking at the raw waveform and then we essentially put a threshold, high pass filter the signal, and then we put a threshold. Wherever that value goes below some threshold, we count it as a spike. That's how we get the ticks. Okay, so everyone happy with the data then? So please play around with it. The more you play, the more things you will learn. It's a lot of fun. I will go back now and tell you the kind of things that we have done with this data set. Okay, so I explained all of these things. So now I will quickly tell you what all we have done with these kinds of. Okay, I don't know why that happened. I will just close MATLAB now. Actually, I'll just exit. I'll come back here. And in fact, I spend a little bit of time here just to motivate, the kind of things that we can do. Am I doing on time? Okay. So power spectral density and time frequency analysis, what I have shown. Functional connectivity, I will get to when we discuss the EEG part. But in the last couple of years, two postdocs, Bhargav and Pooja, and an undergrad, Vignesh, actually use this very data set, the one that you guys have now, to look at some very interesting things. So without going into the details, I'll just actually briefly tell you what all they looked at, just to give an idea of what all you guys can do with this data set as well. So this is Bhargav. He was very interested in understanding if there are waves in this neural data. Is there, if there's a sort of, if you look at this in terms of a two-dimensional, space, x-axis and y-axis being now millimeters. Could it be that if neural activity starts somewhere in the middle, it actually spreads, in this case, like a wave? You can see it's like dropping a pebble and looking at how it spreads, or whether it's a static sort of response. And it's a great question to ask in this particular data set because we simultaneously have two waves, the slow gamma and fast gamma. So you can ask whether these gamma oscillations generate waves, and if so, is there a relationship between slow and fast gamma waves? So the math here is actually fairly complex. So I'll skip that and just show you the answer. So this is the data set that we just looked at. We are looking at a time point where the algorithm has detected both a slow and fast gamma wave. So these are simultaneous waves. And what you will appreciate here is that one wave goes this way at the same time the other wave is going that way. Okay, so these are independently moving slow and fast gamma waves at the same time. And if you do it for all the waves that you detect, you will find that, in fact, now Bhargav is even looking at circular wave and expanding waves and sort of non-Cartesian waves. A very exciting project that he's actually worked on. So you can look at that paper and codes as well. You can actually, have your own, if you're interested in something like this, you can look at it, as well. The data and the codes are all on GitHub. So Pooja in our lab was interested in a question related to what is called phase amplitude coupling. And here the idea is that maybe there are some slow waves, like a theta wave here, that can control the amplitude of a faster wave, in this case gamma. So the phase of theta is related to the amplitude of gamma. When the phase you have a trough, the amplitude is very low. When the phase is, when you are at the peak, the amplitude is very high. It's called phase amplitude coupling.
The traveling waves are in one space.
This is the electrode space. We are looking at the LFP. So you can estimate the phase at each electrode and then do a circular linear regression and see if there's a consistency in that. So, and if that magnitude of correlation is high enough, then you call it a wave. Okay. So all the math is there in that that preprint, that manuscript. Okay, so cross frequency coupling is another thing that you guys can also look at. Her codes are also on GitHub, where essentially this is what we test using some math that I won't get into. Previously, people have shown, so this is how cross frequency plot curves look like, that the phase of theta, so this is about 6 hertz, is generally coupled to the amplitude of gamma. This is called theta gamma coupling. And this shows up as this increase in this coupling exactly at this range. So we wanted to ask if their gamma also couples to slow and fast, gamma also coupled to something. So she did that. So there is a theta gamma coupling in our data as well. This is the same data that you guys all have. So you can actually do this analysis and get a plot like this. In addition, this slow gamma couples to a frequency range between 80 to 150, while the fast gamma couples to a different range, which is between 150 to 500 hertz. Again, what these things mean is there in the paper. and you can discuss this. There are lots of artifacts also that show some of these things, and we have very clever signal processing algorithms to actually deal with those artifacts. This is a very sort of, these are fairly challenging signal processing questions. So once you go through the paper, you will see the nuances about this. So I'll finish off by telling about Vignesh's stuff. So Vignesh actually was an undergrad, like many of you, and then this was his grant proposal. So he said, okay, I'm going to look at bursts of gamma oscillations, because even though it looks like one continuous wave, it's actually not. You get that continuous looking wave because you have averaged over many trials, 35 trials. If you look at an individual trial, you see that gamma comes in bursts. So it evolves and then it goes down, and it also varies in its frequency. So it has a very sort of stereotypical shape. So it's bursty in nature. And figuring out those bursts is a very non-trivial process. Okay, so you need to have very sophisticated algorithms to be able to characterize these bursts, and they have these durations that are, that have a particular probability distribution. And what Vignesh was able to show is that slow gamma has longer bursts than fast gamma in both the monkeys. The distribution also becomes different with a higher proportion of longer bursts. You can see that the blue is above the orange here. And interestingly, even if you sort of equalize the power of these bursts, so you are not, you're saying I'm only going to consider electrodes for which slow and fast gamma have the same power, even then the same thing holds. So it's not that you get longer bursts because you have more power in the signal. Even for power matched signals, slow gamma has longer bursts than fast gamma. In fact, what does it mean? Might ask. So Vignesh actually had a very nice model that he created where they have an excitatory and inhibitory population which are talking to each other. And all he did was change the time constant. So when the time constant is large, the rhythm is at a lower frequency. If you change the time constant, it becomes fast. But interestingly enough, this change is sufficient to actually increase the uh, burst length. Is burst length here, this one is sufficient to increase the duration. So essentially, if you slow down a rhythm, it also becomes longer, which was not intuitive, but that's how the modeling works out. So essentially, a slower rhythm will also be longer if it obeys this sort of an excitatory inhibitory mechanism that we are proposing. Okay, so these are the kind of things that we can do with this sort of a data set, and really encourage you to get going on one of these projects sooner rather than later. It's much less fun when coursework is a lot and you don't have that much time. So in Vignesh's case, for example, after figuring out this slow and fast gamma burst in this LFP data, he is now looking at whether meditators in our EEG data set have longer bursts also as compared to age and gender matched controls. So whether this coordination across brain, which some people say is because of these waves, is more sort of over longer epochs in meditators and controls. There are lots of these analysis methods that you can apply both to the LFP data that you have as well as the EEG data that we'll cover in the next two classes. Okay, so please spend time playing around with this. It's a lot of fun. These projects usually take, in Vignesh's case, it took about a year or so. So by the time the course ended, he had a sort of generally good idea about what he wanted to do. But wrapping it up in 4 months is hard. You'll need a little bit extra time. So those of you who want to take something that like this up can work on it over the summer or so. That's typically the time frame, 6 months to a year for a project of this sort. All right. So we will meet again on Wednesday and we will continue with now the EEG data set. Yes.
What does that imply in terms of most cellular neurophysiology
So what does it imply?
at a cellular?
At a cellular level? Well, so the easy implication is that somatostatin network is slower. Its time constant is larger, which obviously explains why the frequency is low. And it can also mean that it's coordinating over a longer time scale, over a larger spatial extent. So it tells you something about how these different inhibitory systems might be affecting the pyramidal network at different spatial and temporal scales. That's the easy sort of interpretation of this. Very good. Yeah, so if you set this up as a limit cycle, it will just have an infinity long rhythm. So you actually have to inject noise. And we tried a bunch of different types of noise, Poisson noise, it didn't work, Gaussian white noise didn't work. Then this was the work of this PhD student, Krishna Kumaran. He found what is called Olstein Uhlenbeck OU noise. It's a Gaussian white noise that you filter using a low pass filter. And essentially, that will make this rhythm bursty. And it's very interesting that if you play around with the time constants of this OU noise, you can actually either make it go like this or can make it go like that. So actually, his main result was that the inhibitory system is slower because that would explain both the burst characteristics as well as the frequency precession that we see in this, where it starts off at a high frequency and then slows down. And we actually did a clever experimental manipulation where we were able to invert this. So then the gamma actually went like this rather than like that. So that we did in the monkey. That was an actual experiment. So that was his thesis. Um, Again, in all our papers, the codes and data are freely given. Code certainly, data we usually hold on to for some time, but you have now the data. So you can actually run, if you are interested in something like that, you can do that also. Lots of different things that can be done. All right, let's wrap it. On Wednesday, we'll start with the EEG data now. One second, I'll just sharing.